Hello everyone. Today our topic is backcross method. Introduction. What is a backcross? The F1. It is the resultant of a cross between parent 1 and parent 2. When this F1 is crossed with any one of its parent is called as backcross. If this process of backcross is repeatedly used in the hybrid and the progeny in the subsequent generation is called as backcross method. Requirement for backcross program. As in case of every breeding procedure, we require two parent. It is parent 1 and parent 2. But in case of backcross, it is named as recurrent parent and donor or non-recurrent parent. As the name suggests, recurrent. It is used repeatedly in the backcross program. Non-recurrent, that is, it cannot be used repeatedly. Just it is used as a donor parent. So, the recurrent parent should lack one or two characters and it must be the most popular variety of the area with high yielding ability, desired quality and high adaptability. So, this donor parent should have the intense form of character which is deficient in the recurrent parent. So, the intensity of the character should be high and it need not to be a high adaptable variety or high yielding variety. So, the character that is selected for the transfer should be highly heritable and it must be governed with one of few genes. A sufficient number of backcross should be made so that the genotype of the recurrent parent is recovered completely. In order to achieve a successful transfer, we require 6 or 7 backcrosses. Applications of backcross method. Intervarietal transfer, that is a backcross program which is carried out between the species. Interspecific transfer, a backcross program in which the character is transferred from the related species into the cultivated species. So, it is mainly used for the simple inherited characters like disease resistance, seed coat color and quantitative characters like plant height, seed shape and seed size. Next is recovering a transgressive segregant. Here a small modification is done in this backcross method where the F1 is backcrossed to the recurrent parent for 2 or 3 times, or sometimes you can use 2 or 3 recurrent parent to be backcrossed with the F1. So here the different genes, that is heterozygosity of genes are accumulated, results in the production of successful transgressive segregant. Production of isogenic lines. They are identical in genotype except for 1 or 2 genes. So it is called as a near isogenic line. So it is very easily produced using this backcross method. Next is germplasm conversion. It is called as a conversion program. That is, we are producing for example photo-insensitive line from a photosensitive germplasm through backcross method. So here the germplasm is converted in order to produce for a successful breeding program. So it could be possible by using this backcross method. Next is transfer of cytoplasm. So backcross method is used to transfer cytoplasm from one variety or a species to another. So particularly to produce the cytoplasmic male sterility. Here the cytoplasm to be transferred is used as a female parent, that is the cytoplasm which is male sterile which is to be transferred is used as a female parent, and the recurrent parent where this cytoplasm is to be transferred is used as a male parent. So after 6 to 8 backcrosses, the progeny would have the nuclear genotype of the male parent and the cytoplasm of a female parent. This is known as a transfer of cytoplasm by means of backcross method. Genetic consequences of repeated backcrossing. Reduction in heterozygosity. As we said before, it is used in the production of near isogenic lines, that is it will be nearly homozygous in condition. So it results in the reduction of heterozygosity. Next point is increased similarity with the recurrent parent. As we use this high yielding and high adaptable recurrent parent to be repeatedly backcrossed, So the resultant genotype would be similar to the recurrent parent with changes in one or two genes. Next, selection for the gene under transfer. So efficient selection should be carried out in the breeding program in order to stop the allele to be replaced from the recurrent parent. Next is extended opportunity to break undesirable linkage. So in each and every backcross generation, there may be a possibility of crossing over to takes place between the gene being transferred and the gene that is tightly linked. Next is selection of recurrent parent type. So the selection of recurrent parent type will lead to faster replacement of genes from the non-recurrent parent. Procedure of backcross method. As we all know, in order to carry out a crossing program, we need two parents. So in case of backcross, we are using a recurrent and the non-recurrent parent. In this diagram, the recurrent parent is an adapted cultivar A and the donor is a resistant cultivar. So when this cross between the disease resistant cultivar and the adapted cultivar A results in F1 and this F1 is backcrossed to cultivar A, it results in the production of BC1 progeny. So in this diagram, you could see that they have mentioned that percentage of genes from A. As it suggests, if you go for repeated backcross, a desirable homozygote increases rapidly. That is, the genes from the non-recurrent parent are rapidly replaced from the recurrent parent. So as a result of continuous backcrossing, the progeny would be identical to the recurrent parent except for the gene that is to be transferred from the donor or the non-recurrent parent. So as a result of this backcross method, the resultant variety would be identical to the recurrent parent with high yielding ability and high adaptability and also with the character of disease resistant which is obtained from the donor parent. This backcross method is of two types which is based on the triad that is transferred. Either it may be dominant and it may be recessive. So in this case, if the F1 hybrid is crossed with the dominant parent, that is a dominant donor parent, so it produces 100% tall progeny. But if it is crossed with the recessive donor parent, it would result in 50% tall and 50% dwarf. So let us see in detail what is a consequent of this 100% tall and 50% tall and 50% dwarf, which makes the difference of carrying out the backcross procedure. Transfer of dominant gene. Here the donor parent is rust resistant and the recurrent parent is rust susceptible. So as we know, it is a dominant gene, that is capital R and capital R, and the susceptible with small r. When this both are crossed, the F1 would be heterozygous for rust resistant. Since this capital R is dominant over small r, all the plant will be heterozygous with rust resistant. And when this F1 is backcrossed with the recurrent parent, it would have a half plant would be resistant and the remaining half would be susceptible. So here we are selecting the plants which are resistant and then we are allowing it to backcross with the recurrent parent. So this procedure is continued till B6 generation where the similarity of recurrent parent would reach more than 98%. Next in the after backcrossing for 6 generations, the individual plant progenies are grown, that is they are homozygous for rust resistant and similar to the recurrent parent, and then they are allowed for the replicated yield trial along with the check variety to predicted performance for the yield and other required characters, and then it is released for the variety. Transfer of recessive gene. The donor parent is rust resistant variety with the recessive gene and the recurrent parent is rust susceptible with the dominant gene. So when this both are crossed, it produces an F1 generation and it is backcrossed with the recurrent parent. In the BC1 generation, rust resistant is recessive. So all the plants are rust susceptible since it has a capital R in the heterozygous condition. So the plants are allowed to self-pollinate. Then the completely resistant plant with smaller genotype is recovered and it is allowed to backcross with the recurrent parent. So this procedure is continued till 5 or 6 generation. Then the individual plant progenies are grown and the check varieties are grown along with it to determine the rust resistant genotype, and the selected genotype is released as a variety. Advantages of backcross method. The genotype of new variety is nearly identical with that of recurrent parent except for the genes that is transferred. So the outcome of this program is known before, so it is very helpful to reproduce at any time in future. Next one is only a smaller population are needed than the pedigree method. It is not necessary to test the variety developed by backcross method in extensive yield trial since the performance of the recurrent parent is already known. So it saves time for nearly 3 to 5 years and also reduces the expenses. Next, defects like susceptibility to disease of a well-adapted variety can be removed without affecting its performance or the adaptability. So, the variety which is very popular among the farmers will remain popular after this backcross program. The backcross program is not depend on the environment since this off-season nursery and greenhouse conditions can also be carried out for advancing 2 or 3 generations. It would drastically reduce the time required for developing a new variety. It is the only method which is used for producing the interspecific gene transfer of cytoplasm and also for producing the transgressive segregation. Disadvantages of backcross method. Superior varieties cannot be produced except for the character that is transferred to the recurrent parent. Since by repeated backcrossing with the recurrent parent, it may result in nearly 98% similarity with the recurrent parent. Next is linkage drag. As in case of this backcross, some kind of crossing over may take place between the transferred gene and the linked gene, which may result in the linkage drag. And the hybridization is to be carried out for each and every backcross, which is difficult, time consuming and costly. Thank you.